thank you for joining me because the work you've done is quite interesting, and there's two major pieces of work that I think we'll discuss, as well as sort of the overview of the areas that you've worked in or you work in now. But the first thing is really sort of the major component is you've looked at a lot of mRNA and sort of, lipid nanoparticles. Was that what your PhD was on? So take me back to when sort of you started working with these systems, or what was your PhD on?
Yeah. So when I started my PhD, I was really intrigued by drug delivery in general. I like the idea of sort of building these carriers that can take drug molecules to where they need to be. And so with that in mind, my PhD was on nanotechnology and on drug targeting to tumors. So how can you get a drug molecule only in the tumor, but nowhere else in the body, so that you can get maximal efficacy, maximal activity of that drug in the tumor, but not in healthy tissues, right? So that you can reduce side effects. So that's what my PhD ended up being on. But the piece that I sort of took from there was really nanotechnologies and how you can develop them for a variety of applications. I no longer work on drug targeting as such, but I use these nanotechnologies for delivery of biologics and mRNAs and so on now.
Yeah. The final bit you said there, the mRNAs and biologics, they've really taken off, especially since COVID-19, where, we realize the value of them in vaccines and treating diseases, and they've also taken off in other areas in terms of inflammation and cancer and all sorts. What do we mean when we talk about sort of mRNA delivery? What is it exactly?
Yeah. So I think I'll try to explain it from a very simplistic sort of an eighth grade level, and then we can build up on it. So all our cells in the body have proteins, and proteins are really our workhorses. So whenever a cell needs to do something, say for example, if it's a muscle, if it needs to contract, or if it's a cell that's trying to move from one place to the other, it always uses these proteins as its workhorses to do that business. Unfortunately, when these proteins don't work, that's when we get diseases. And so if you can change the expression of proteins, you can really impact a lot of disease areas. And for the longest time, we've been using chemicals, or what we refer to as small molecules, to modify the expression of these proteins, or to change either how much there is of it, or how little there is of it, when it sort of comes about. And we use these small molecules to change their expression. But sort of moving forward now, we've realized that there are other ways in which we can modulate the expression of these proteins. And so we take a leaf out of the biology textbook here, and we look at the sort of the central dogma of biology, which is that cells in our body have DNA molecules, and protein really comes from those DNA molecules. But our cells are very stingy when it comes to how they want to make these proteins. And they don't sort of keep making these proteins all the time, but they want to make it at only sometimes when they actually really need it, right? And so that's where the messenger RNA comes into picture. So this messenger RNA is that intermediate between the DNA and protein, which is expressed only when we actually want that protein to be. So that messenger RNA is temporarily expressed, while the DNA is there permanently, right? And when that messenger RNA is expressed, it then makes the protein of interest. Now, sort of going back to what I said about how proteins modulate diseases, you can change the expression of proteins now through DNA, or you can do it through mRNA, and both of these are sort of legitimate ways of doing it. The reason why people are interested in messenger RNAs is because of their temporary nature. So because they are expressed only for short periods of time, for which they express the protein, this becomes very attractive from a pharmaceutical, from a therapeutic standpoint, that you are expressing whatever protein you need only for a short period. So once the person is sort of treated, that protein is no longer perhaps needed. And so you don't really need long-term expression of that protein. And that's where messenger RNAs come into picture. You get it in the cell, it makes the protein you want, and then it's degraded by our cells very rapidly. So really, that temporary nature is attractive. But because they are so sensitive to sort of degrading mechanisms within the body, making drugs out of them is very difficult, because as soon as you put them in the body is going to destroy it, right? So
Yeah.
how do you protect the messenger RNA before it makes the protein? But then as soon as the protein is made, it needs to go away. And that's where a lot of the bioengineering and formulation development techniques come in. Um, that sort of focus on the chemistry of the RNA as well as chemistry of the nanoparticles then.
So one of the papers you published, and actually the first paper I read of yours, was on sort of radio protection
Yeah.
against radiotherapy and patients that have gone through radiotherapy but need protection in terms of the oral areas as well as the sort of prostate and the sort of that area of cells because they become degraded and damaged, due to radiotherapy. So I thought that'd be quite nice to bring that up as well because it segues quite nicely into the use of nanoparticles. So that paper itself had a lot of novelty, I think. There was a few things in it that was quite unexpected that you wouldn't normally see. So we'll come on to that in a second.
Mhm.
What I wanted to know is how did that research come about?
I was in the best place a researcher could imagine. And so, what I mean by that is that at the time I was working with Professors Bob Langer and Gio Traverso at MIT and Brigham and Women's Hospital, Harvard Medical School. Um, and these guys have sort of put together a team of individuals who are just coming from very different backgrounds. And so in that sort of uh, playground, if I may, one of the individuals I met was James Byrne, who is a radiation oncologist. And so with him, we started thinking about some of these toxicities of radiation therapy, as you mentioned, and how can we sort of better manage them. At that time, I was already working on messenger RNA therapies to the gastrointestinal tract. And really, that's how we sort of came together. And we thought that, hey, it'll be really interesting if we sort of learn from other organisms as to how they protect themselves from high radiation toxicities and perhaps apply that through messenger RNAs into human cells. So I think it's a sort of a shout out to team sciences and putting people with very different backgrounds together who have an open mind to discussing ways of doing different things.
I think that's such a nice way of thinking of it, and it's good that you was in and that sort of mixed environment because when you're in a team of people that think very similar to you, you're not necessarily going to have that cutting edge mentality because you're all thinking similar. But actually, if you're in a team where you're with a radiologist who's thinking about the patient first and seeing what
Mhm.
he's seen in clinic, but you're coming from this pharmaceutical background
Yeah.
with nanoparticles and drug delivery, it's a nice pairing of the two.
Yeah,
And I
it's
think
not just sort of clinical translation. I think that was sort of I thought that that was the biggest learning that I had from that experience. But also, when you have people from outside of your field, they challenge you, not to challenge you, but to know more about how you plan to accomplish XYZ, right? And as they ask you those questions, and as you're saying things out loud, you perhaps realize that your sort of chain of thought is not perhaps as airtight as you would have thought it would be, right? And so when you start speaking with people outside of your field, you just, or when you get challenged by people outside of your field, I think it just sort of pushes you more to do better.
so. Okay, So you've got this paper, right? And correct me if I'm wrong. Again, it looks at how radiotherapy damages different areas. So it looks at things like head and neck radiotherapy. And one of the things that I've read was that it can be such an issue because the radiotherapy can cause patients to take a break from the radiotherapy, right?
Right.
Yeah. And then that causes like failure or decreases the chance of improvement, right, from that radiotherapy.
Exactly. Yep. Yep.
Okay.
So yeah, so I think we specifically sort of focused on two areas in that paper. You're exactly right, that people who have, say, for example, head and neck cancer or prostate cancer, they can be treated with radiation therapy. And honestly, I mean, radiation therapy can, in these cases, give you excellent outcomes. But nearly all the patients who are undergoing radiation therapy experience side effects from radiation therapy. And so it's because radiation therapy doesn't really distinguish as much between healthy cells and cancer cells. So as it's trying to kill these cancer cells, it also kills off healthy cells. And so because of that, these people get something called mucositis, which in the mouth it's called oral mucositis, and for prostate cancer patients, you see that in the colon, and that's called colitis. And in both of these cases, you're basically having these healthy cells die off because of radiation therapy. And so with people with oral mucositis, they are unable to eat very well. So, and without that sustenance, they're not strong enough to take radiation therapy. And so it sort of forces the clinical team to either reduce the dose or to reduce the number of cycles they're going to do. And as you can expect, if you do that, the tumor, the cancer has an opportunity to sort of return. And so the overall efficacy of the therapy goes down. So our expectation here was that we develop a system that can protect healthy tissue, thereby allowing the clinical team to sort of use radiation without any sort of breaks from it.
Yeah. And in the process of you looking at these mechanisms to sort of prevent that break, you focused on this little animal in the sea, the tardigrade. How did your attention end up on these species? So yeah, how did you come about this?
Yeah. So tardigrades. So just so people know, this right here is what a tardigrade looks like. It's obviously not to scale. They're supposed to be about a millimeter or so in length, and you can see that's 8 feet. It's kind of cute. They're called water bears. So these are some of the most radiotolerant organisms on Earth. And they've been sort of subjected to quite a lot of damage, and they've still sort of survived it. For example, they've been taken out into space and open to space, and they've been able to survive the extreme radiation seen over there. They've been taken to very high temperatures, very low temperatures, and they still survive. And interestingly, they've been sort of shot out of a gun to get them to have sort of a lot of physical mechanical stresses on them, and they've still survived it, right? So that's why these are these extremely tolerant organisms that are quite amazing. So the question is that why are they able to do this, right? And so they haven't really evolved to survive radiation. That was not sort of the point of why they are that tolerant. Really, the idea there is that they live in environments that undergo a lot of fluctuation. And so when the environment around them is not hospitable, these organisms undergo desiccation so that they can live in low resource environments for a very long time. And they get to sort of water contents of about 2 to 3%, right? So we are about 70%.
Yeah.
A lot of organisms are about 70%. These guys get to about 2 to 3% when they are exposed to those environments. But because or they get to these low water contents, the cells are experiencing a lot of stress, and they get these extremely high levels of reactive oxygen species in them. And so, how do they protect themselves from these reactive oxygen species? So to do that, they've started making a lot of proteins that protect their DNA, their mitochondria, their cytoplasm, etc., from those reactive oxygen species. So we thought that what if we take one of their proteins, put it into human cells? Would that enable us to protect human cells from reactive oxygen species? And these reactive oxygen species are caused when you expose cells to radiation. And so that was sort of the whole logic as to what we were trying to do. And to deliver that protein, instead of using the protein itself, we used an mRNA that encodes for the protein. And remarkably, we found that if in fact we make that protein in human cells, it protects human cells from radiation damage. And so that in itself is quite interesting, I think, because tardigrades, as you can see from their shape and structure, they're not like us. They're not our cousins. They're not macaques, right? They're very different from us. Um, uh, but despite that, the proteins that you find in tardigrades are actually working in our cells, which is remarkable as to why that happens, but it happens, right? So, yeah.
When we say lipid nanoparticles, what in this context specifically, what is the structure of your lipid nanoparticles? Because you have liposomes, you have solid lipid nanoparticles. What exactly did you use?
You mentioned three types of lipid-containing nanoparticles: liposomes, solid lipid nanoparticles, and lipid nanoparticles, right? So the way I think about it is that liposomes are typically this monolayered or bilayered structure where you have the polar heads of the lipids facing towards the aqueous side and the nonpolar tails sort of coming together. Giving rise to sort of two compartments in that liposome, one that can house the hydrophobic drugs and one that can house a hydrophilic drug. Solid lipid nanoparticles, I more think about them as a matrix-like system where it's sort of like a chocolate chip cookie, if I may, where the dough is essentially these solid lipid nanoparticles, and the chocolate chips are the drug of interest for you. In contrast, lipid nanoparticles, and I should mention that the structure of these lipid nanoparticles is still under quite a lot of debate. In lipid nanoparticles, we think about it as somewhat similar to liposomes in that you have these the heads of the lipids towards the outside, and then you have hydrophilic pockets on the inside. But these hydrophilic pockets are lined up by positively charged lipids. And so because of that, you can put in your negatively charged RNAs or DNAs in these little hydrophilic pockets. And that allows you to make these carriers that can then deliver the RNA. These are not just made out of these positively charged lipids, but they also have three other components in them which allow for their folding and structure and their stability then.
I see. So that charge helps to maintain the mRNA within its sort of vesicle so that when you deliver it, it brings it with it, and it can also sort of increase that entrapment within that
Exactly.
system. Yeah.
Exactly.
Okay.
Yep.
Okay. So you've got these systems, you've got these lipid systems. By which mechanism are they then moving into cells? How are they able to sort of move into cells, but then also release that content into the cell?
Right. Right. So the idea here is that RNAs they're very large, millions of Daltons in size, negatively charged. So they don't really diffuse past cell membranes as small molecules would. Now, when we put it into nanoparticles, we are actually making them physically in size, we're making them larger, right? Because now it's not in sort of this angstrom range. It's about 100 to 200 nanometers in size. But when we entrap them in lipid nanoparticles, we change the route of cell intake from diffusion to endocytosis. So from a passive transport to an active transport mechanism. And so these lipid nanoparticles enter cells through endocytosis, and they enter into these regions called endosomes and lysosomes. Um, Now, a major problem in the field right now is how do you get these lipid nanoparticles out of the endosomes and lysosomes and into the cytoplasm, where your RNA can then interact with the ribosomes so that it can then be translated into the protein of interest. But the components of the lipid nanoparticles are designed so that they can destabilize the endosome and lysosome membrane. And upon that destabilization, the RNA is sort of expulsed into the cytoplasm. Having said that, I should say that current estimates sort of put it between 1 to 10% of the RNA that's inside the endosomes and lysosomes makes it to the cytoplasm. That number is, again, I think it's very fluid. I don't know how accurate that is,
Yeah.
but what we know for sure is that it is bad. It's very poor. And so there's quite a lot of efforts at improving that.
Building off of that then, so focusing on the endolysosomal escape mechanism, one of the things you did within that research was you introduced polymers, right?
Mhm.
What was the logic of that? So,
Yeah.
we mentioned that conventional sort of lipid, but then you also added the polymers as well.
Yeah, it's a great question, and I'll sort of go a little deeper into the mechanisms of both lipids and polymers, and it'll sort of come together then. So, as I said, the way these lipids work is that they once they are in the endolysosomes, these lipids interact with the lipids membrane of the endolysosomes, and they destabilize them, breaking them apart. On the other hand, polymers, and these polymers have really been used from the 90s, and some of these mechanisms have also been sort of derived in the 90s. With the polymers, the idea is that once they enter the endolysosomes, the endolysosomes are really acidic in pH, and the polymers we use have an alkaline pH to them. So once these polymers go in, they start buffering the pH of the endolysosome. So the pH comes up. Now the cell tries to actually get the endolysosomal pH to drop again. And in order to do that, hydrogen ions from the outside of the endolysosomes enters into the endolysosomes. And as hydrogen ions enter, it draws in water along with it. And so that gets the osmotic pressure to rise inside the endolysosomes, and they get them to expand and then ultimately burst. And so this is called the proton sponge effect, because it's these protons that are coming in that drives the water in, and that gets these endolysosomes to burst. So sort of taking these two things together, the lipids are destabilizing the endolysosomes through interaction with the endolysosomal lipids, while the polymers are doing it through a proton sponge effect, right? And so if you go back to your undergrad pharmacology classes, when you have these two drugs that are working through different mechanisms, you have the potential of sort of combining them to give you better activity. And so we use that sort of pharmacology logic here in drug delivery, where we said that, hey, if we combine these two modalities together, we may perhaps have both mechanisms of endolysosomal escape, thereby giving us better RNA delivery. So that's how we thought about putting these two things together.
I see. So you was hoping to bring two different mechanisms together to increase that escape, because as you previously mentioned, the escape is 10 to 20%.
Yep.
But we're trying to raise that by adding another mechanism to hopefully, break that endolysosome down so you can get more escape.
Exactly.
You tested a lot of polymers within that paper, within that research. I think it was over 200 cationic polymers.
Yeah,
And I think in the end, you ended up using two polymers that you tested
right.
to within the research. What things were you looking at when you were testing these polymers? How did you firstly come up with these 200 polymers? And then what about these two polymers really stuck out for you to say, let's take these further?
Yeah. So in sort of to your first question of how did we come up with these polymers, um, the logic here was that we needed positively charged polymers because we were complexing negatively charged um, nucleic acids, and we needed that sort of basicity or alkalinity in the polymer. And so, because of that, we use these amine-containing polymers. So the you can, if you think about this polymer, uh, you you can imagine them to be having an amine in the backbone, and then sort of these amines are connected through little carbon chains. So what we do change is what hangs off of that amine, and then what is the carbon chain that separates these two amines from each other, right? And so by changing those two things, you can make essentially a library of polymers. And that's how we went to about 200. The point is that we didn't know what we needed to sort of get a very good transfection efficacy. That's why we take these sort of unbiased approaches where we essentially test 200 of them, 300 of them, and see whatever works. Whatever works, we then take forward, right? Sort of a medicinal chemistry approach to pharmaceutics. Um, Now, what did we test for? So, given that our ultimate interest was in transfecting or delivering mRNA into the oral epithelial cells to protect against oral mucositis and the colon epithelial cells to protect against colitis, uh, what we asked was if we just use the polymer to deliver the mRNA, which of these polymers delivers the mRNA best? And so, based on that analysis, we picked one polymer for the oral epithelial cells, one for the colon epithelial cells, and we hypothesized that if we have this one polymer that is by itself doing very well at transfecting these cells, if we put this polymer into the lipid nanoparticles, it'll boost the activity of the lipid nanoparticles the best. And so that's how we chose these two polymers to sort of dope into the lipid nanoparticles.
I see. So you was looking at the different areas that they could target and what was performing the best. Okay. The final sort of bit, I think, where the part of the paper as well as the research that was quite interesting was actually looking at the results
Mhm.
of or the impact of this sort of mRNA transfection, but the impact on the models you were testing.
Yeah.
So how did you go about testing the sort of the efficacy of the delivery within living organisms?
Yeah. So, are you talking about the protein from the tardigrade,
Yeah,
or are you talking about just sort of a model mRNA?
the mRNA from the tardigrade once that
Yep.
was loaded. So how did you measure
Yep.
that?
Yeah. So we did a sort of two or three levels of testing on that. So as I said that these tardigrade proteins, some of them protect their DNA from radiation damage, and so we express those proteins in human cells, and then we expose them to radiation, and we measured how much damage is done to the DNA in cells that were not transfected with the tardigrade protein versus cells that were transfected with the tardigrade proteins. And so these are fairly um, sort of common assays for people who measure DNA damage. For those who are interested, it's called a gamma H2AX assay and a COMET assay. It essentially measures if there are breaks in the DNA after it's been exposed to radiation. So that was sort of an in vitro study that we'd done. We then took it a step forward and we said that, okay, if we actually are reducing DNA damage in these cells, are they able to multiply better then? Because remember, mucositis arises because you're killing off cells in these various tissues. And so we did sort of a standard proliferation assay. It's called a colony forming assay to see whether these cells are able to actually grow or not. And so all of that sort of yielded quite promising results. And so then we tested that out in mice, where we injected these mRNAs in the cheek area of the mouse, and then to that area we expose it to radiation, and we look at DNA damage in the cheek. Again, comparing mice that were not treated to the mice that were treated, and sort of what we see is that you get protection in the area that the mRNA was applied to. So for example, if you applied it to the right cheek, we get protection only in that area. And in the left cheek, we wouldn't get protection then. So it's a highly localized protective effect that we see with these therapies.
And what about the duration? So, once you've injected them, how long do you have up until you can then do the radiation?
Yeah. So in our present analysis, we've seen that it's about 6 hours or so. So 6 hours after mRNA therapy, we see maximal protection. There is some protection that we see as early as 2 to 3 hours as well. But certainly it's maximal at about 6 hours. By 24 hours, it wanes off. This almost perfectly tracks our pharmacokinetic data that shows that protein expression also follows the same trend. So it increases by 3 hours, 6 hours is maximal, and then it falls off by about 24 hours. Now, one can look at this as an advantage or a disadvantage. An advantage of this is that you're not really getting these body cells or these human cells, excuse me, to have these proteins sort of permanently expressed in them. But the disadvantage is that it's actually a very short duration. Individuals who are undergoing radiation therapy, really, they have multiple cycles, and it goes on for weeks together. So in its current form, it'll be really hard to apply this in the clinic if you're injecting the patient with these mRNAs 6 hours before radiation therapy each time, right?
Yeah. And I think there's something you mentioned as well towards the end of that research was, as just a small statement, you mentioned something about looking at the formulation type. So, at the moment it's injections, but you mentioned looking at topical formulations, microneedles, and I think that could help with some of the things you mentioned, the challenges of that long term, which is quite a nice, I think, way of looking at things. What impact do these have? So they're obviously they're protective molecules, right? Or protective formulations to protect against the radiation. What effect do they have on the cancer itself?
Yeah, that's a great question. So in theory, if this was to go into a cancer cell, very likely it will protect the cancer cells against radiation damage as well. And so mechanisms that can allow us to get them away from cancer cells are critical. This technology cannot work without them. Okay, so now how do we sort of think about that piece? I think there are a few parts to this. The first part is that you sort of strategically pick cancers that are at anatomically distinct areas from the organs that you want to protect. For example, in prostate cancer, the major toxicity is in the rectum. And the reason is because as the radiation beam goes through, it first hits the prostate and then the rectum is right next to it. Okay. But anatomically, when you're trying to treat these tissues, you won't treat them at the same place. If you put something in the rectum, it's not going to go into the prostate, right? So although for the radiation beam, they're next to each other, from a pharmacological standpoint, they're not really connected to each other. So that makes it a very attractive sort of a cancer area where you can apply this into, which is where you just have anatomical separation between the cancer and between the organ that you're trying to protect. That's one aspect of it. And this is relatively straightforward, I should say. The other aspect of it is technology building, which is far more complicated, which is how do you actually make technologies that allow your mRNA to be targeted to healthy tissue. This can come about by first making devices that can specifically target healthy tissues. For example, can you 3D print microneedles that are complementary to the contours of the tumor so that you're only applying the nanoparticles in a certain area? Second, can you make nanoparticles that are selectively taken up by healthy cells and they're not taken up by tumor cells, which is sort of the opposite of a lot of research that's going on right now, right? But nevertheless, that's the sort of the fundamental idea there. Number three, can you make RNAs that don't have the mechanisms for protein expression in tumor cells, but that do possess mechanisms for protein expression in healthy cells? Now, all of these on paper sound like ideas that can be sort of put into practice, but they're much harder to do because boundaries between healthy and normal tissue are not that stark, either from an anatomical standpoint and also from a biochemical standpoint. And so really targeting healthy tissue while sparing tumors is much harder to do. Hopefully we come up with good ideas to do that, but right now we haven't shown it.
Yeah, no, I think the first thing you mentioned there was like the pharmacological selectivity, because if you're, just because something is covered by the radiation in terms of the prostate and the sort of the rectum, it doesn't mean you can't, because you mentioned previously, if you inject something in the left cheek or the right cheek, you do not get protection in the opposite.
Exactly.
So you can still have that selectivity. And you mentioned something there is the second thing you mentioned was it's kind of the opposite of what we're doing, because in terms of targeting the healthy rather than the cancerous tissue. And it's true because when you look at nanoparticles and cancer drug delivery, it's always the EPR effect, for example,
Right.
where we look at the pH or we look at the permeation of the cancer cells. But as long as there's something differentiating the two environments, that means the formulation may have some ability to differentiate as well. So,
Right.
as you mentioned, there is an opportunity there. The whole tardigrade experiment was very nice because I think there's so much in nature already that
Yeah.
we can learn from. And I think it's very easy to look past it because when science is going in one direction, I think a lot of people end up following that direction. So you'll see a lot of things that are very, very similar, but actually, if you pay attention to what's going on around us, there's a lot of answers already embedded within nature. And I think it's a very nice bit of work.
Yeah. Thank you. Thank you very much.
Thank you for joining me. I appreciate it. And I think the work you did was very interesting. Like I mentioned, both of the papers, fantastic work. And thank you.
Thank you very much for having me. Appreciate it.